Hello friends, this video on microbes in human welfare part 6 is brought to you by examfear.com. No more fear from exam. So now let us look at microbes in industrial products. Now, as I had mentioned before, that microbes are not only useful in the household products, but it also helps in producing a lot of products on a commercial scale, on a large scale. Now, what are some of these products? So some of these are antibiotics, which is going to be extremely important because antibiotics are those medicines which help to fight severe bacterial infection, and this is used for the treatment of several diseases like typhoid, diphtheria, and so many other bacterial diseases. So and here we get to know that it is microbes which help in the formation of antibiotics. So antibiotics are also prepared with the help of microbes. Next is the beverages, like the different types of alcoholic drinks which are available in the market. Wine, beer, everything is prepared again from microbes. Enzymes. Now, enzymes are extremely important. Now, when we say enzymes, we are not only talking about enzymes, but there are various chemical substances which help in so many different processes. And many of these enzymes are prepared from the microbes. So that way also, and all these things can be prepared from microbes on a larger commercial scale, and that is why they fall under the category of industrial products. Like for preparation of all these items, it is not that somebody can prepare antibiotic at his or her home by taking small amount of microbe and following the process. Now all these things, they need a lot of machinery, a lot of setup. So these are performed on an industrial scale, that is on a commercial large scale. So let us first talk about antibiotics. So now whenever you think of antibiotics, you might remember somebody who would have fallen ill and he or she would have got treated with antibiotics. So antibiotics is an extremely useful thing for our welfare. And these antibiotics are prepared from microbes. That is so interesting. Microbes help in preparing antibiotics. And what is the role of antibiotics? To kill other microbes. That is to kill disease-causing microbes. Because antibiotics are effective against bacterial infection. So how is it effective? Because it kills the disease-causing bacteria by breaking the cell walls of the bacteria. So basically prepared from some microbes and kill some other disease-causing microbes. So that is the purpose of antibiotics. So what are antibiotics? These are the chemical substances which are produced by microbes that kill disease-causing microbes. So that this statement itself proves that there is one set of microbes which are useful, there is another set of microbes which are harmful. So antibiotics will kill the harmful microbes and they are prepared from the useful microbes. Now, these antibiotics were discovered. The first antibiotic was discovered by Alexander Fleming, and he was a Scottish scientist. And the first antibiotic was penicillin. Now, it was named after the microbe from which it was extracted, and that is why the name was penicillin. So we will talk about its discovery in the next slide. Now, if you look at the name antibiotic, what does it mean? So anti means against. So if you look at the word anti means against, and biotic. So what is bio? Bio means life. So antibiotic, the term means against life. So what does that mean? Because it is against living organism, against the disease causing microorganism. So that is why it is against life. But if you think of yourself, like if you think about human beings, it is not against human beings. It is pro-human beings because it helps human beings to live. But it is against the living organisms which are harmful to the human body. So that is why the name antibiotic. Now let us quickly look at the discovery of penicillin. Now this discovery again is very interesting because it was discovered by chance. what we know call as serendipity. So whenever something is discovered just by chance, so Alexander Fleming was not performing specific experiments to discover an antibiotic. He was doing some other experiment with a bacteria, and it was just by accident, or it was just by chance, that he discovered that there is something called antibiotic. There is something which is there in the living organism that can kill the bacteria, and that is how penicillin was discovered. So penicillin was a chance discovery, which is also termed as serendipity. So anything which is discovered by accident or by chance. So what was he doing? He was actually experimenting with a type of bacteria, that is Staphylococcus. So he was doing some experiments with this Staphylococcus bacteria. So while performing this experiment, what was he doing? He was growing this bacteria on a petri dish. So petri dish is nothing but a cell culture dish. And what did he observe? It was observed that once it so happened that a petri dish was left open by mistake. So it was not covered and it was just left open, and it happened by mistake by Fleming, and he observed that it got contaminated by blue-green mold. So here you can see some mold growth. So this entire plate where the culture is taking place, this is called petri dish. So this is how Staphylococcus bacteria were growing. But when by mistake it was left open, it was found that some mold growth started taking place on one side of the petri dish. And this mold growth was quite visible. And the most important thing which was observed was that the mold contamination on the petri dish inhibited the bacterial growth. That means it didn't allow the bacteria to grow. So if you see, so this is where the mold growth takes place, and in the surrounding of the mold growth, there were no bacteria which were able to grow. So no bacteria were seen in the vicinity of the mold contamination. So what did this prove? So there was something very important which was concluded out of this observation. So his conclusion was that mold released a substance that inhibited the bacterial growth. So there was, it didn't happen by magic, right? So there has to be something in the vicinity of the mold growth that resisted the bacteria to grow, that stopped the bacteria to grow. So he said that mold released some substance, and that substance inhibited the bacterial growth. And that substance today we know as antibiotic. So he named that substance as penicillin after the name of the mold, that is Penicillium notatum. So this mold which was growing here, this mold was Penicillium notatum. So from that name he named the first antibiotic as penicillin. So this substance was nothing but the antibiotic, and antibiotic was nothing but penicillin. So this particular antibiotic was named penicillin. But later, over a period of time, many different antibiotics were formed from different other microbes. So, but this was the first antibiotic. So this is how penicillin was discovered. Thank you. Please visit examfear.com for an easy 4-step learning process absolutely free of cost. Watch video lessons, ask questions, refer notes, and take an online test. Thank you once again.